Hello, and welcome to our single-cell multiome analysis series with SciDAP. In the previous tutorial, we started the dimensionality reduction analysis for RNA. Today, I will show you what the output of the pipeline looks like and will give you a few tips on how to reduce the batch effect even more precisely. There are a few tabs with the data. Let's start with the quality control. There are 3 plots. Let's look at the first one, the elbow plot. It represents the number of principal components that we chose for our dimensionality reduction analysis. Or you can say this is the number of dimensions, set as 40. You could see it wouldn't be much difference if we would choose 50 or 40. This is how you assess if you're in a good range. Now let's look at the correlation between QC metrics and the principal components. There are the most important ones: RNA reads, genes, mitochondrial percentage, and the novelty score. For these two, there is not much variation. However, for RNA reads and for the genes, we have some of the variation. But it is good that the correlation coefficient is not too high or too low. We see our 4 UMAPs that were produced during the RNA dimensionality reduction. Visually, you could already distinguish some clusters, but their analysis would be our next step. Now we only have our QC metrics that are plotted on the UMAP. Then you could switch to the next tab and see more plots where cells are colored by dataset on the UMAPs. And also, they could be split by the datasets in the different UMAPs or whatever number of samples you have. This information is useful to assess whether any of the clusters might be enriched with the cells from either of the donors. In that case, it could indicate strong batch effect, which we could correct by rerunning this dimensionality reduction analysis, changing the parameters for batch effect settings. I will show you later in this video. Their QC metrics split into 4 different grades: low, medium, medium-high, and high. And these UMAPs picture the RNA reads per cell, there is a genes per cell, and the mitochondrial percentage. Then, in the next step, you could see the same information, but split by the condition. If you remember, we had 2 groups: the recirculating cells and the tissue-resident memory cells. To explore the batch effect settings I was talking about, let's clone the sample first. And here, where we were providing the batch correction method by Harmony, we provided the group for batch correction as a dataset. It means that it will be correcting the batch effect between every sample, because every sample is a dataset in our metadata. Now, let's say we don't want to do it for every sample, but only for donors. And for this, we need to change our metadata. New metadata file could be found back at the filtering tab. Navigate to the Files tab and search for the metadata file. Here it is. With a Microsoft Excel, you can open it. There is a column which will have the information about the donor. It has to be named Custom Patient, and make sure that it corresponds to the files that were uploaded and filtered in this pipeline early in the same order. And you click Save, and go back to the experiment to set the batch correction parameter to the new custom patient parameter, which corresponds to the column that we just created. And here you provide your new file. You click Attach and select the sample, then Run. In the next video, we will start the next step, the RNA clustering.